Good morning, everyone. I'm Tony Lovegrove. I lead business development at Discovery Life Sciences for Northern Europe. Really delighted to have you here today. A number of my colleagues I will hand over to soon. I've got a few notes that just warm up by way of preamble. Just wanted to say, the most enjoyable aspect of my role as business development lead for Northern Europe are the days when I'm away from the desk, as I have been all week, often travelling in tandem with a variety of my colleagues here, connecting with folk and hearing about the stories, the blueprints, and the toils of bringing a life to projects that we have the privilege of being part of the formative discussions for. And yeah, that really is one of the most enjoyable aspects of my role. Always had a high regard for the life science membership organisations and ecosystems that we enjoy here in the UK, drawing a huge amount of energy from their investor events, their networking and scientific sessions, and especially the summer party, the OBN summer party that now seemed a long time ago. I can remember going over to Hammersmith for that event, I think in June, Liz. So it seems a long time ago. So they're excellent events. And so with the support of my manager, who's sitting at the back there, Oliver, Marta, John, Nadia is not here, she's in Boston, especially our subject matter experts, Annetta, Jane, Don, and Hugh from QIAGEN. We conceived today's Lunch and Learn in collaboration with OBN. It feels good to be here. I'll feel a lot better, though, when I pass the mic over to Jane, who will get you underway. But before I do, please allow me to remind you to be sure to remain after this talk, because there'll be lunch laid on here at the back of the room and the opportunity for networking. And also, as the speaking wraps up, there'll be a session of about 15 minutes for Q&A, and we'd be delighted if you could interject with the speakers or amongst yourselves for that Q&A session. So that's all from me. I'm going to hand over to Jane now. And before I do, just to recap on today's agenda. Okay, thanks. Over to you, Jane. Thank you.
Thanks, Tony. Well, can I just say that the weather compared with yesterday's liquid sunshine in Manchester here is far better. So I'm got hats off to you in Oxford. This is wonderful. So I'm going to talk about the context of diagnostics and getting a therapeutic to market in that context. So my background is I'm a companion diagnostic expert, which is one way of saying I've been around the block a bit. So what are we talking about here? We're talking about biomarkers, companion diagnostic development, the right laboratory partnerships to get both the therapeutic and the diagnostic to market, IVD manufacturing, and the appropriate technology platforms that you would need to get a diagnostic to market that could support a drug. And there's a very standard way of developing companion diagnostics. You can see it here. It follows all of the normal workflow that you would see with a therapeutic. So you have a preclinical phase where you develop your diagnostic in feasibility, a phase 1, 2, and 3 clinical trial, and then you submit for regulatory approval. It's exactly the same process that you see for getting a therapeutic to market. And when you're doing this, you're usually doing it in conjunction with a pharma partner if it's a companion diagnostic. So you'll have a joint steering team, and they'll work very closely together to get that drug and the diagnostic approved together and get regulatory approval. So what does that mean? Regulatory approval in the FDA, in the US, you've got the FDA, and in the EU, you've got the EMA. But there's a little known almost loophole with the EU approval. So it's called Health Institute status under CE-IVDR. And this is the equivalent of LDT, the laboratory developed test in the US. So there's a way that you can get a diagnostic out there being used in support of a therapeutic without going through all of the performance trials that are required. So this Health Institute status is a very useful way if you're really pressurised for time. As I've said, it's very important to get biomarkers integrated into your therapeutic. So that means developing good strategic partnerships with diagnostic companies and also understanding those market needs, and the commercialisation and reimbursement landscape is always very important. So the challenge, what are we trying to do here when we're trying to get diagnostics and drugs to market? You've got a regulatory challenge, you've got the diagnostic development and the drug development, you've got 2 clinical trials which are sometimes combined, often are combined, across 2 different organisations, both the drug and the diagnostic, and it makes it incredibly complicated and incredibly complex. So what happens? Your timelines are inevitably squeezed. There's always a rush to get things done and achieved. As I mentioned before, this Health Institute status is a real advantage for CE-IVDR regulation. It means that you can conduct those clinical trials without requiring the full CE-IVDR license, and it means you can skip some of the performance studies because basically the EU are saying we trust this institution, it's got the correct regulatory approval and accreditation with us. And it means that you can benefit from much faster EU approval, and if you've got the right samples in place, you can get your drug and your diagnostic to market much quicker. How much quicker? Well, a lot quicker. So pretty much you can halve the timeline it would take to get your diagnostic to market, and you can also pretty much halve your costs as well. So it's a dramatic improvement if you use this route. If you're working with a partner, you would need the correct support. And what sort of support would you need? You'll need to have ideally an agnostic, platform agnostic approach. You need end-to-end support, both for your research use end phase of your diagnostic and for the LDT phase. And you would also apply a multi-omic approach to get your therapeutic to market with the right diagnostic. All of these things help, and access to samples. So what you need to have is this global infrastructure and regulatory alignment. So CAP Clear, GCLP, ISO 15189, that's what you choose. And a single project manager to unify everything under a unified LINS system. So in summary, I'm going to close out now before I hand over to our next speaker. But in summary, what you need is multiple platforms available at the drop of a hat. You don't want to have to install a platform base. Health Institute status is going to get you there much faster. And samples are always an issue. So if you've got a biorepository, that's always a real advantage. And the ability to align those regulatory requirements across various different territories will save you a lot of time and money as well. So thank you very much, and I'll hand over to our next speaker, who's Don. I think you're going to...
Thank you, Jane. It's a great intro to topics that we'll be covering today, really from a high level from the beginning to the end, and so now we'll go into much more depth. I'm Don Skifter. I lead our scientific affairs in proteomics and genomics at Discovery Life Sciences, and I'm really excited to be here today. Thank you all for showing up. Thank you to Tony for setting all of this up, and to the organizers for helping put on a great event. So thank you. So I will focus on really the upfront part of when you're headed towards developing a companion diagnostic or other diagnostics. What are the important factors to think about for pharma sponsors, for biotech, to think about as you're beginning to move towards a CDX? I think you may need a CDX with a particular drug. And so I'll talk more about the discovery aspects, the exploratory aspects, and those considerations. And then the following speakers will really get into the details of healthcare institute status and the details around development of CDX. So you've all seen these slides for many years now, right? But we're really in the era of precision and individualized medicine, which is for me, really exciting. I finished my PhD in 2003 on the heels of the completion of the Human Genome Project, and what a great time to enter this space, right? But we've come so far, but where we're at now, we're just on the cusp of what we're ready to do, and I think in personalized medicine. So just as a quick review, of course, it used to be we'd develop a drug, hopefully, we'd apply it to any individual across the population, and it would work in a subpopulation. You'd need that to get approvals, of course. But then there were those who either there was no efficacy, or worse, you'd have adverse events. And those are, of course, the things we want to avoid. And that's really what precision and individualized medicine is doing for us today. We're able to treat a much larger population, or at least the relevant population, based on biomarkers and based on CDX that we're able to generate, and hopefully avoid treating those who won't have any efficacy, or certainly those who would experience adverse events. So we look at the history. CDX approvals have been most common in oncology. In part, assays like using immunohistochemistry has been around a long time, very usable. It's well known. Pathologists like IHC, and so it's great testing modality, right? And then came next generation sequencing, and of course, their DNA variants that told us something about development of oncology drugs. So that whole history exists, but now we're really, with the new technologies that are coming out, the scale of those technologies and the quality of the data we're able to obtain, both in invasive original site tissues, but even in less invasive tissues or biofluids like blood and plasma, we're on the cusp now of really taking this into so many other areas. And some of this has obviously been done, but so much more is going to happen very soon. So I could personally say I've been working in our proteomics division the last couple of years, and the number of immunology and neurology projects that we're working now on biomarkers and discovery and exploratory work to discover those biomarkers, to take them in some cases down the CDX path, it's really increasing. And again, the technologies are enabling that. So that's mostly what I'll focus on in the next few slides. And, I mentioned here immunology, neurology, but think about GLP-1 inhibitors, right? They're everywhere now. They're being utilized a lot, but there's still some issues with them. There's side effects. And so there are biomarkers we may be able to identify to help with that and better treat in those conditions. Infectious disease to avoid adverse events. All of these areas we are now working with biomarkers. So what are those modalities? Of course, many of you know about DNA and RNA sequencing at the bulk level. We can now do that at the single cell level, both at the protein and genomics. Using flow cytometry for target engagement, receptor occupancy to better understand what's going on, immunohistochemistry, which I've mentioned, spatial transcriptomics, and really exciting in the bottom right here is just a differential expression plot for proteomics. So in our, we often use Olink or other affinity-based measures, and even now mass spec, where from a discovery and exploratory-based perspective, we're able to detect 10, 15,000 proteins in a given sample, depending on the sample type. So from an exploratory discovery perspective, the technologies are really enabling now with really high quality data. And of course, to follow on the heels of this right now is, of course, artificial intelligence, machine learning, which has frankly already been around for quite some time. But the data sets are now getting big enough where hopefully AI is going to make sense of what's really going on when there's missing pieces of the matrices across patient cohorts. So I want to just talk a little bit about a couple of case studies. And here I want to talk about plasma and moving into plasma and trying to identify biomarkers from plasma, which is a less invasive biofluid than, say, going to a primary tumor tissue. And what this represents here is within the plasma, there are a lot of proteins. The top, 90, 99% of those proteins are made up of the top 20 proteins. And so how do you get to those mid expressors and those very low expressors? Well, there's technologies out there, nanoparticles, and taking those nanoparticles to purify or get rid of, deplete those high quantity proteins. So you're able to see the lower expressors. And you can look at that a few different ways then. So for example, if you're looking at interleukins, chemokines, cytokines, that's telling us something about the immune response, hopefully from that biofluid. But then for those biomarkers that may have leaked from tissues into the blood, we're also able to detect those. So it's almost a 2-for-1 in many areas where you can sort of monitor the immune system, what's happening there. And perhaps those are biomarkers of interest, but also from the tissue itself, be able now to identify biomarkers in plasma and other less invasive biofluids that tell us something about the disease state and help us drive towards these CDXs. So it's a really exciting time. And again, these technologies now are really enabling this. So we did a COVID study just to begin to look at what we're able to detect with a couple of proteomic measures. And here I'm showing Olink HT, which is an antibody-based affinity platform. HT that recognizes about 5,400 proteins. And then on the right is mass spec and use of mass spec in these COVID samples. And what you're seeing from these COVID patients is a nice overlap in the proteins that are detected. But it's not a huge overlap, 15 to 20%. We're seeing this across different diseases between, say, Olink and mass spec or other affinity-based measures of mass spec. Great, we have that commonality, but what's going on with those differences? Well, we're able to access different information based on the biofluid or the approach that we're taking. So it's giving us, from an exploratory discovery perspective, a much broader base of what can we actually identify to then look for smaller signatures or even single analytes that you might take into a CDX. And if you look at more of that detail, this is showing the proteins here on the left with the mass spec data in the blue, with the Olink data there on the right that were detected. And again, there's that small overlap, but there's clearly a large difference. And you can see in the signaling pathways that are activated a difference there as well. So they're truly complementary in the data that they're offering to us and that we're able to utilize. One more quick case study here, and again, sticking with proteomics. This is a large oncology cohort that we're working on right now. These were pulled from our own internal biobank, where we're doing FFPE samples from the primary tumors. On that, we'll be doing whole exome sequencing, RNA sequencing, mass spec. And then on the matched plasma samples for a proportion of those patients, being the plasma from the same cancer patient, we'll be also doing mass spec as well as Olink on those, and integrating these data sets to see what sorts of biomarkers are unique and where are they overlapping. And so this is an ongoing project, but I'll present some of the preliminary data. And this is specifically data in the plasma that I'll be presenting here. But once we have we're just about finished collecting the whole exome, the RNA-seq data, we're going to overlay that, and begin to integrate these data sets. So just high level, I won't go through the details of the data, but looking at the differential expression analyses, again, on the left here with Olink Explore HT, and on the right with the mass spec approaches here, the Thermo Fisher Orbitrap Astral, which is an amazing mass spec instrument. You can see we have a whole variety of proteins that we identify that are either up or down regulated. And again, looking at the overlap of those, there is some, but only, again, 16%. So we're getting, again, very unique information from these different platforms in terms of what we're identifying for biomarkers. So complementary in what we're able to see. Interestingly, when we do look at just the overlap, we're seeing the same trends, which is a good thing. The technologies for the overlap are telling us the same thing. Here, increases in expression of a variety of cancer-related proteins pulled from these markers were pulled from a Cell publication, but we looked at our data to say, hey, at least for these, are they increasing or decreasing? They're all at least trending in the same direction. And this theme continues. I presented the COVID data. We're working now in immunology, dermatology diseases, things like atopic dermatitis, psoriasis, et cetera, where we're using, rather than skin biopsies, now skin tapes. And we're able to scrape those skin tapes, pull the proteins out of those, both a soluble portion as well as taking the actual cell pellet, hit it with SDS, get peptides, take that into mass spec. And again, we can overlay those data sets to say, what are we seeing in other differences between those soluble proteins versus membrane-bound or other things we're able to get out of those cell pellets. And again, some overlap, but clearly some unique differences. The complementarity is still there. We're able to do this in urine. This was just the healthy internal metabolism study we did. And then what I'm really excited about, I just came back from the AAIC conference, the Alzheimer's Association International Conference from Toronto. And basically nearly every talk there was talking about how we need to increase the use of biomarkers in neurology, and not only Alzheimer's, but neurodegeneration more generally. And so excited to see what's happening in this space and what we're now identifying. And I'm just super excited about that. And again, just in this case with CSF, again, you can see the complementarity of the approaches. And again, this is very proteomics focused, right? But the genomics, all those other modalities that I've already talked about, we're going to be able to utilize that to get better insights in unique biofluids and tissues that maybe in the past you couldn't, or you want to fit the right technology to the biofluid or the tissue to give us meaningful markers that you can then take into smaller signatures. So overall, then, the biomarker landscape initially kind of looks like this. But with Olake, mass spec, you begin to clarify that picture. Future technologies are going to give us much more. And then multi-omics and ultimately AI hopefully will give us the full picture. So we're able to now go from these very large panels and affinity-based panels to targeted approaches, things like Target 48, Flex, and Focus, which are really small custom panels. You can see on the right some differential expression between the high-level panels and here a Target 48 panel. Same thing with mass spec. The thermostellar instrument that we have is very translational, so you can do thousands and thousands of samples quickly on a targeted set of proteins, but still using mass spec. And the Astral sort of feeds it that information so we know what we're looking for, and it's really for use in translational space. Or you can cross over from technology to technology and do this. So again, just we're enabled so well now to take this much further. So I'll just end by saying, what are the overall considerations that you want to look at? So again, maximizing the relevant biomarker insights, thinking about the tissues versus fluids. Do they really represent what we're looking for with your particular drug? And are the technologies giving you what you really need that's going to tell you what you need for your drug that you're trying to take to a CDX? All while thinking about budgetary constraints, because we can't do it all, right? And so it's really important to pause and think about these strategies up front before you're at phase 2, phase 3, and it's too, maybe not too far along, but you want to start thinking of these things early in discovery. Moving from invasive to non-invasive tissues and biofluids, I've talked a lot about that, but just examples, again, tumor FFP into plasma, we're able to do that now. And I'm really excited, for example, we're starting to now also work with some extracellular vesicle companies to say, can we increase the signal with EVs from the plasma that might be coming from the original tissue as well? So, we'll be able to increase that sensitivity, I believe, as we go. And this is work we're just beginning, so that's me speculating at this point, but I have a lot of hope there. Another one I've talked about is the skin biopsies going to skin tapes, and now we're even being requested to run interstitial fluid from skin to become even less invasive. So still, again, early days on this, but I'm excited to see, the decrease in invasiveness of these different sample matrices. Layering sample inputs, of course, continues to be important. And then also very important, and I think this will be addressed more by Annetta and certainly Hugh from QIAGEN, the kitting and partners that you'd need to work with in that IVD downstream. Who's going to be the long-term kitting partner for that if you would take that sort of an approach? So thinking about that up front. And then your turnaround times, not only for testing on the exploratory side, but the final side, but, we get to drug approval more quickly if you're targeting the right populations with inclusion-exclusion criteria using these biomarkers, even if they're not yet an approved CDX. That's much of the work we do at Discoveries in that space and using them for inclusion-exclusion. And then finally, the Healthcare Institute status, where Jane already touched on the timelines and how they're shortened, but Annetta will speak more to that here shortly. So I will end with that. Again, focus more on the discovery exploratory side, but these are all considerations you need to think about before you even get into your CDX, and look forward to chatting with you all and answering any questions you might have later. So thank you all for your time. This is my colleague, Annette.
Thank you. Hello, everyone. Just a short introduction. I'm leading the Department of Genomics and Molecular Pathology at Discovery in our German lab in Kassel. And the site in Kassel is also the health institution that I want to talk a little bit more in detail about. So I take over, let me see which direction. Yes, so I'm taking over at the point where you discovered one or a few biomarkers and need them in clinical testing. And I want to go a little bit more into detail about the different custom assay development and validation strategies that you have. So one option, of course, is that you can partner early on with an IVD manufacturer, and you will tell you more about this after my talk. The other option that you have with the health institution, especially if you have a clinical study that you have to bring up as early as possible, would be as depicted here. So we list the several options that we provide as a health institution. And as long as the biomarker assay is not needed for patient management decisions, so meaning for inclusion or exclusion in a clinical trial, we can validate a GCLP assay as a lab-developed test for research grade. So this means we are still showing the analytical evidence of the assay, but the testing is not later on used in a clinical setting for patient inclusion criteria or exclusion criteria. then our first go option would be, of course, to use a commercially available CE or US IVD assay that is already available on the market. Because in this case, we only have to do an assay verification, which is the fastest to bring up an assay for clinical testing in a clinical trial. However, I have to say, these assays are rarely available for new biomarkers or for the specific needs in a clinical trial or for the patient population. So what to do then? So if patient management decisions are required for the biomarker in the clinical setting, you can either, of course, go the whole way into a performance evaluation study. But again, as you heard before, that if the timeline is really critical and you have a clinical study start as early as possible, another option is to validate an assay as an in-house IVD. And for this, we utilize our full IVD-capable equipment, and we also use our GCLP CLIA workflows that we have established in our lab in Kassel. And during the IVD in-house test validation, we show a clinical evidence as well as the analytical evidence for the validated assay. So, and this gives the option to use this biomarker assay for patient management decisions in a following clinical study. Let's go to the next slide. I want to go a little bit more into this specific option of an health institution status and the validation of an in-house test now. So again, you hear it all the time, again, is the timeline and the cost efficiency of using an in-house test, simply because it's really important. And we see that the timelines are very often constricted from the partners that we have and the customers that do contact us. So it enables a faster and more efficient assay development and validation timeline. Of course, it's also more cost-effective compared to going a full performance evaluation study. And the most important thing for you is probably that we have a faster study start up compared to doing a full performance evaluation study. So, but now I want to go a little bit more into the applications and examples of use for an in-house test, because I think this is sometimes a little bit unclear. So where can we use the option of an in-house test? So, of course, you can use it for a custom assay development for new biomarkers. And another option would be to validate an existing IUO assay that is commercially available already, but not validated to the level where you can use it for patient management decisions. So in this case is another option where you can use an in-house test validation. Another important use that we do very often, I have to say, it enables an off-label use of an existing IVD assay that is commercially available. And examples for this are, for example, that you need an assay that is validated for another indication, or it's bridging to another platform. So very often the intended use of an available IVD are very constricted for a certain use. And with the validation to an in-house test, we have the option to open the intended use a little bit more up, so to redefine the intended use, and then use this assay with the background of the technical validation that was already done for patient management decisions. And of course, lastly, it's if you want to have like the IVDR security documentation and want to validate a secondary endpoint biomarker. So this would be also an option where you could use in-house tests. I want to mention other relevant aspects. That is, for one point, that we always can rely back to the technical validation that the manufacturer of an assay already provided. So we do not have to start from zero unless it's really the development of a new biomarker assay. And another important thing is the availability of biospecimen. And here also our partners and customers become very important because, of course, we can rely on our very big biobank for assay validation. But it's also helpful if the customer can provide biospecimens that are as close as possible to the clinical study later on. And this is also a point where we can really accelerate the timeline further and also the cost efficiency of assay validations. So I want to even go more into detail and give you real-world examples of actually ongoing or just finished in-house test validations that we did in our site at Kassel. You are probably all aware that HLA genotyping became really relevant in clinical studies for showing immunotherapy response prediction. And in this case, there are IVD kits available on the market that are commercially validated for patient management decisions. However, they are restricted in the intended use for the purpose of using it in the context of transplantation purposes. So in this case, we did repurpose the validation with the in-house test, and this enables us to use them for HLA genotyping for immunotherapy studies. Another example is that we validated and targeted NGS-BRCA panel, and in this case, it helped us to guarantee the continuous use during the whole duration of the study of the available IVD assay, because of the manufacturers have to get ready for IVDR. So not all IVD assays that are currently on the market can span the duration of the full clinical trial that can span 3 years, for example. So by validating this assay as an in-house test, we have the option to guarantee that we can use the same assay without the need for a bridging during the whole clinical study. And another example is, for example, HPV genotyping, which is normally used for diagnostic purposes for cervical swabs in women's health, became relevant in clinical trials. And here we did a bridging and repurposing of existing IVD assays related to the sample type. So in clinical trials, very often FFPE tissue is required as testing material, and the intended use of the available IVDs was restricted to cervical swabs. So in this case, we can repurpose the in-house test by opening up and doing additional validation for FFPE tissue. And of course, lastly, but that's also very important, is the de novo customized assay development and validation, for example, for RCL PCRs, that's all related to CAR T-cell therapies, where RCL detection is needed as a secondary endpoint for safety measures. And then we also can offer gene expression assays for custom target genes and also SNP genotyping assays. So you heard a lot about the orange part depicted here in this slide, about what we can provide as a service from discovery. And now I want to hand over to you, who will tell you much more in detail about the CDX route when you partner directly with an IVD manufacturer. Thank you.
Okay. This one is. Thanks, Aneta. Thanks very much to Discovery Life Science for inviting me to speak today and yesterday. It's a pleasure to be here. My name's Hugh Ricketts. I'm part of the business development team at QIAGEN. I'm going to assume everyone knows who QIAGEN is, right? Everyone's seen the blue and red boxes. Maybe you don't know what we do in terms of partnering for precision diagnostics. So I'll go through a little bit of that today, particularly about the commercialization of a diagnostic assay linked to a drug, which would be a companion diagnostic. I'm going to cover kind of two things, actually, and I've got 3 points on here. I think Aneta's shown you really nicely in early phase clinical development of your drug. It can be very costly, time-consuming. I think James said that as well. And coming to an IVD provider like QIAGEN, we do that regularly, but it can be time-consuming. So if you don't have time before you start your trial, it can be challenging. So what Aneta has shown you is a really novel and simple way to get ready for clinical trials, at least in the EU. So I won't go too much into that first step. Well, what I will go into is what happens later on in clinical development once you're getting close to drug launch. How do you actually have an IVD that can be commercialized and support the prescription of your drug? And having that companion diagnostic on the market doesn't always necessarily mean the patients will be tested and drug will be available. Just so you know, this picture on the left, Jane, the sun always shines in Manchester. I don't know what you're talking about. It does that. This is QIAGEN Manchester, our building on the hospital complex in the centre of Manchester. And we moved in there about 3 years ago. State-of-the-art laboratories. It's 5 floors. We have all 5 floors of that. About 300 QIAGEN employees based there. This is where we do all of our companion diagnostic development across our platforms and other. You'll see other company platforms as well. Bit of context of QIAGEN's companion diagnostic pedigree. This is what we like to call our trophy cabinet across the top. We've been in Manchester actually developing companion diagnostics since about 2009. We acquired a company called DXS who had some technology around PCR technology around KRAS and EGFR mutation detection. QIAGEN acquired them in 2009. About 50 employees at the time. We've grown that now to 300 employees. And across the top, you can see our companion diagnostic approvals that we've had during that period, starting all the way back in 2012 with Eli Lilly and their KRAS. Actually, it's an exclusion criteria for the drug Rebtux. All of these are PCR-based assays for oncology. So most of them tissue-based, also 1 or 2 liquid biopsy-based, PI3 kinase there with Novartis. So we like to think these are all kitted assays available globally, wherever you want to buy the tests, it is available. So we QIAGEN consider ourselves to be the number one kit provider for companion diagnostic development. There are diagnostic developers out there who have more single site-based assays. Tests, Guardant Health being one of them for liquid biopsy in the US, Foundation Medicine as well. But QIAGEN can distribute these kits globally. So consideration when you're developing a drug is, can you actually get that test available to as many patients as possible? A kitted solution, in theory, should enable some of that to happen. We have more than 35 ongoing pharma partnerships. At any one time, around 40 companion diagnostic development programs. We have the global regulatory experience for all global territories, so FDA, IDR, Japan and China. We're able to develop assays across the globe. Our clinical and medical affairs teams are there to support that enablement of our tests in clinical trials and then obviously further commercialization. We have third-party agreements with technology providers such as Illumina. So we have an agreement with Illumina that enables us to develop IVD assays on the Illumina system. And then we're developing new technologies internally as well. So digital PCR is a key one there. We're trying to look for some ultra-sensitive protein detection on that platform as well, which would follow on nicely from what Ron showed with more of a discovery if you're boiling that down to a few targets. So on our technologies, we have quantitative PCR on the RGQ, the Rotogene Q, which is standard PCR, and also a QIAstat system, which is an enclosed sample purification and PCR system. We're across hematological cancers and solid tumors. And the QIAstat, actually, we're starting to roll that out into non-oncology as well. So we have an agreement with Eli Lilly to develop an ApoE4 genotyping test for Alzheimer's. Digital PCR is one of our critical platforms. Sensitivity, precision, really important. And as I mentioned, we can use the Illumina IVD systems to create sequencing assays. So we can do all the technology. We can hit the requirements of development, and commercialization is then critical. So we can roll these tests out across the globe using I'll come to this in a little bit more, but using a network of our lab partners. So wherever that drug is, the test can go as well. So just to kind of follow on from what Annette was telling you about development of a diagnostic assay, this is fairly simplified, although it looks fairly complicated, but a simplified chart of how long it would take QIAGEN to develop an assay, probably for a later phase clinical trial, but it could be early phase as well. And where we would need to build out the evidence, the analytical evidence that assay does what it's meant to do prior to putting that into a patient in a clinical trial setting. The challenge with that is it does take around 18 months to develop the data that's required to support any regulatory submission. And this is where Annette was talking about the Health Institute in Europe. The Health Institute status really supports a reduction of that timeline. What we see the biggest timeline really is the submissions to the European authorities and the notified bodies across each different country to submit the analytical data to get approval to use that in a clinical study is really quite time consuming. But when you come to later phase development, you really need to start thinking about going through this if you want to have a kit that's available globally, because these are the type of studies that will need to be done prior to launch of your drug or prior to using it in a pivotal study. So once you have your drug on the market, there are a number of reasons why patients may never get tested and then therefore never get a targeted treatment. This is a nice paper from 2022 where the authors looked at in advanced non-small cell lung cancer, so quite heavily personalized, quite heavily precision medicine orientated non-small cell lung cancer. EGFR, KRAS have been around for a while now in lung cancer, PD-L1 as well. They looked in a cohort of non-small cell lung cancer patients, how many patients you start with who are diagnosed with non-small lung cancer who actually get a targeted treatment at the end of it. And you can see here that they started with 1,000 patients, all diagnosed with non-small cell lung cancer, and actually only 356 of those patients got the targeted therapy as they went through that journey. And there's a number of gaps, as you can see here, showing that there's reasons why they don't get therapy. Not wanting to have a biopsy, I suppose that's a patient decision, painful, invasive, dangerous in some cases. The specimen was not collected correctly, or the pathology, the evaluation was not performed correctly. Those gaps, number 4 and 5 there, this is really where companies like QIAGEN can really help, in that maybe physicians don't know how to order a test, don't know the test is even available. The test is not potentially reimbursed in different markets, so they never order the test for the patient. Or once they do order, the test performance is weak, you don't get a valid result. Or when it is reported, the physician doesn't understand the report and isn't able to actually action that and put the patient onto therapy. So it's a little bit of a depressing view from a precision medicine perspective. Precision medicine is obviously meant to support more patients getting on treatments that are actually going to benefit from. But commercialization of IVDs and commercialization of testing can be challenging. That's why you need to partner early with companies like QIAGEN so we can actually get testing into the market, educate physicians. So hopefully I can show you a little bit of that now. What we do at QIAGEN is really start very early with our pharmaceutical partners. And part of the reason that maybe in non-small cell lung cancer that didn't happen is we didn't partner early enough with certain partners. So what we tend to do is anywhere between 6, but probably more like 12 months before the launch of a drug with a companion diagnostic, we're sitting down with a pharma partners, building a plan of how we're going to have this testing available. And we use a network of labs that we have agreements with across the world where we can implement our technologies prior to launch of the drug and make sure that those tests are going to be available day one of drug launch. So this is kind of tier 1, building that base, as we say there. Once the drug is launched, we can then start disseminating the test out to other labs who want to take up those tests and to broaden that coverage in key territories or across the world. And then tier 3, really rolling that out further into more decentralized testing environments. I think the key thing for QIAGEN is that we're able to do this because everyone knows what blue box is. There's not many molecular labs out there that don't know QIAGEN, so we have excellent view on where testing is taking place and where it is needed. You can see here just 3 aspects of commercialization of an IVD. Marketing. You saw on a previous slide that there could be points where physicians don't even know about a test. They're not educated about it. So marketing is really about educating those physicians, understanding the patient journey, understanding where testing should be done and at what stage in their journey that should be done. So we start that very early in IVD commercial launch. We would start that at least 2 years prior to launch for an IVD. Reimbursement, actually getting it paid for, is challenging. It's different in every country, very different systems. If it's a novel, brand new biomarker, it can be extremely challenging to get good reimbursement. And labs won't test if they're not getting paid for testing. So critical piece of work that's done, again, a long time before launch of the drug and the IVD. And of course, the medical affairs aspect of it. The beauty of companion diagnostic development from an IVD perspective anyway, is you're implementing that into a drug trial. So you're creating the clinical evidence that assay is relevant in that particular patient setting with that drug. But you may want to build that evidence to convince physicians that this is a good idea to have their patients tested. And that's that medical affairs piece, is building that clinical evidence to educate physicians and also to get broader adoption of the test. So I mentioned our our day one laboratories. We have a network of these. It's actually more than 30 labs now across the globe, including China, Japan, etc. All different markets, all different ways of testing. Japan and the US, for example, very highly centralized testing environments, whereas in Europe, a little bit more decentralized. Samples tend to stay in countries or regions. So we can meet all those kind of testing requirements through our network of laboratories. So just to finalize, I think QIAGEN, our precision diagnostic development and commercialization, our goal, really, our vision, is to be able to work with pharmaceutical companies to develop these tests alongside the drugs, to be able to globally distribute those so that patients can get access to testing. And our mantra really is, wherever that drug goes, then the test needs to follow. We can do that with various different technologies. We can do that in partnership with Discovery Life Sciences, for sure, where we can implement our technologies in their laboratory to speed up particularly early phase development. If you do that, that reduces risk as things are transitioned into a more commercial environment. That just gives more options to our partners, that depending at a particular phase of development, depending on the requirements, on the market requirements, there's different models to partner with diagnostic companies and laboratories. In terms of the whole idea of this really is to be able to deploy different diagnostic requirements into different markets as they need that, and build that diagnostic strategy early on with our pharma partners. And that really ensures ultimately success in market access, being able to guarantee the patients will be tested. So that's it from me. I think I'm handing over to John, and then happy to take questions later on. Thank you.
Hi, everybody. Let's see if I can get it right. So I'm John Beck from Discovery Life Sciences. My role here is to manage our strategic account and opportunities, also our CRO and China partners, and also our peri and post-approval services. And there's a strong partnership theme to my role, and I think that really represents well, I think, some of the key takeaways, certainly that we at Discovery we have from today. And I hope you've really enjoyed this session as we come towards a close. I'm the one between you and either lunch or going back to work. So that's either a good or a bad thing, I'm not sure. We wanted just to share a few of our own takeaways, but your role, your company focus, that of course will determine what you take away, and we certainly hope you found this useful. Jane opened setting the context here, and I hope not too alarming, and I'm sure most of you are aware already, there are many, many challenges. This route, this ecosystem, the landscape we work within, it's complex. And I think, through partnership, through working together, through networking events like this, and then establishing and furthering those partnerships, these challenges are surmountable. Certainly we believe this, and we work hard to achieve that. after lunch, if you want to talk to the team, we're all going to be here and start to look at any of your unmet needs or any of your challenges. We're very happy to do so. Don then opened the biomarker discovery end of this spectrum, and really it's a continuum. And I think the take-home message there certainly is that orthogonal testing adds huge value. And I've got so many technical questions around how you deal with this, because we didn't cover the digital space, we didn't cover AI. I mean, this is an ever more complex and bigger data sets that we need to make some sense of and have the power and understanding from a biology perspective to really get to the right marker that then you take forward to implementation in the patient setting for precision medicine and ultimately for partnership with IVDs like QIAGEN. And that's where I think very well took you through some of the scenarios and some of the, again, challenges and thoughts around implementation of and now a more targeted biomarker set, or even a single marker, a single SNP. Even we see a lot of usage these days. It's not all complex, it's not all small or large, medium, large panels. It's individual singular biomarkers. But consider all the options is the takeaway. There are many options. There are, look at the partnerships that you can achieve. Look at orthogonal testing, but decide with the end in mind. Consider where you want to go, and then have that as your marker for the timeline. Even this is the biggest challenge I think we see: timeline. Honestly, even more than cost, I would say. And then Hugh, I think, very well took you through a lot of the thought processes from one of our best IVD partners within the molecular space. And I think there is partner well. You will absolutely rely upon their expertise, their pipeline, to get you to your endpoint. And I think engage early. More and more, you need to be thinking, even in that early translational space, actually, how do I reach the patients that I want? How do I get that drug and the CDX to the right region geographically? Super important decisions, but the earlier you make it, and the better you partner with your IVD, I think, the more success you'll see. I'll stop there. I've just have this one slide. I hope you have your own takeaways. I hope some of these, resonate well with you. It's just left to me to say thank you, really, because, your time is valuable. We appreciate everybody being here. We appreciate your attention. So thank you so much. We're almost at Q&A, and then beyond that, lunch. But maybe if you'd just join me in one final round of applause for the speakers, I think, and I hope I've done a good job for them.